Okay, good morning, everyone. We shall discuss about transposons, where we started yesterday. And to start with, I will just have a quick review. We are talking about something very interesting that is about genes that can move from one place to another place. That's an amazing thing, as if it's an if it is as if it is a dog or a cat moving from one place to another place. Imagine a DNA molecule being able to do that, somehow making its own copies at different locations within the genome. That's something amazing, right? How did it evolve? And why did it evolve? And why is it still existed? These are some questions that will border with philosophy. So, in a way, all of biology is philosophy. And I think you have to develop this philosophical inclination to trying to know what is truth and not, and trying to know what is the purpose, or how things happen and why do they happen. That's up to you. You can educate yourselves on it on YouTube. If you want, you can always contact me if you need further help in that. But what we are talking is about transposons, and there are three different classes. In the last class, we learned about a lot about DNA transposons, right? And there are two others, which are virus-like retrotransposons and poly-A retrotransposons. The first one is named DNA transposons because they are just DNA molecules moving from one place to another place. The other ones are called retrotransposons because during their movement from one place to another place, it's not the physical movement, but it is actually the copy of that transposon that is moving to a different location. And during that copy is made using or made as RNA. And from RNA, DNA is made, and that DNA moves to a different location. That's why they call them as retrotransposons. Some of those are viral-like, and some of them are special category called poly-A retrotransposons. I hope you remember what is autonomous, what is non-autonomous. Autonomous transposons are those that have all the elements required for transposition to occur. What are all the elements? There are only two special or essential things in a way. One of it is the border or the terminal inverted repeats. The second one is the gene for transposition, because the enzyme is the one, transposase is the enzyme that binds and moves the transposon to a different location. Say if some transposon does not have its own transposase, then you call it as non-autonomous, because then it is dependent on a helper transposon. That is a transposon somewhere else which could supply the transposase enzyme. There is something else called grounded. It's just a name to say that if the inverted repeats are broken or got mutated, then they cannot move any further, and then they are said to be grounded. So in today's class, we'll also discuss a little bit about what we have studied towards the end of last class. And I hope you also remember what is TSD, right? Target site duplication. So transposase recognizes the border inverted repeats, and then makes a cut and then moves it to a target site. During that site, it makes two cuts, not at the same location, at two different locations like this, because of which you will end up having target site duplication. Okay, I hope you remember we have discussed all this mechanism of how things happen. The retroviral, before we have to appreciate what the elements that are present in retrovirus, like viral-like retrotransposons. You have long terminal repeats, and then you have gene for integrase and reverse transcriptase. So what it does is the long terminal repeat on this side has the promoter as well. This is the promoter. Using that promoter, it is transcribed and you would generate an RNA molecule. That RNA molecule can be translated, and you will get reverse transcriptase. You can also get integrase. Okay. And what else can also happen is that the RNA can be reverse transcribed by the reverse transcriptase to produce complementary cDNA or complementary DNA. Okay, I mean double-stranded one, and then it is processed by integrase to it will remove these small fragments, and that 3' OH is then makes a nucleophilic attack on the target DNA and gets integrated, something like this, right? So, and then the gaps are repaired and ligated, and you might end up having these target site duplications. So what are the important steps? Every step is within here itself. If you know the genetic organization of a retroviral or these transposons, then you can say everything else about it. It has long terminal repeats. It has gene for integrase. It has gene for reverse transcriptase. What is the function of reverse transcriptase? To generate double-stranded DNA from RNA. And what is the use of integrase? It processes that cDNA, double-stranded cDNA, and integrates it into a target location. So everything else is in the genetic organization itself. Okay. Then we are seeing the next category, that is the poly-A retrotransposons. You call them poly-A retrotransposons because they have a poly-A and they are involving a mRNA molecule. So you call, or RNA molecule, so you call them as retrotransposons. So yeah, the architecture wise, it has 5' UTR. UTR is untranslated region. And you also have 3' UTR, that is the 3' untranslated region. So upon transcription, okay, let me finish this. There is a promoter on the 5' UTR, like in the previous case too. Then there are two open reading frames called as ORF1 and ORF2. So upon transcription, mRNA is generated, and upon translation of that mRNA, you will get ORF1 and ORF2 proteins. Those proteins will form a complex, and they bind to the mRNA, the same this one. And they will kind of scan the DNA, and if they find any AT-rich region, something like this, right? Remember, this is a poly-A retrotransposon. It has so many adenines in this direction, so many of them. So when you generate the mRNA will also have those poly-A. So ORF1 and ORF2, they come along, they kind of scan the DNA, and because they can, this T can, if you have multiple T's like this, they can be base paired with the mRNA, which has a poly-A tail-like thing, right? So they will bring in the RNA here, and then they will kind of base pair it like this. They'll make a nick on one side somewhere here, and the poly-T is base paired with this poly-A. And now the first cDNA strand is synthesized because you have a poly-A. I mean, the template is the mRNA here, and then you have the first cDNA strand synthesized. After that, once this DNA is synthesized, the second strand is broken somewhere like this, and then it is the sequence that is present here is copied and made into a new one here. Not everything is well understood, but they have deciphered many steps, but there are some more things as to what are all the things that are involved. It's not completely known. So what you are ending up is having an ORF, this poly-A retrotransposon that is present in one part of the organism genome is copied, is made into RNA, then reverse transcribed, and then you have it got integrated, right? That genetic information got integrated. So that is kind of, it is jumping genes. Copy of that information is moving from one place within a genome to a different genome. That is about poly-A retrotransposon. So you try to remember the structure of it. You know the 5' UTR, 3' UTR. There is a promoter here. You have ORF1, ORF2, and everything else is here. Poly-A is after the UTR, right? What is the use of 5? The promoter will synthesize the mRNA. ORF1, ORF2 are the ones that bind to it, scans, make a cut, and allows or promotes the synthesis of first cDNA strand. What is the significance of poly-A tail? Poly-A, that poly-A is the essential one to decide which is the target site. Basing on the poly-A, it is going to base pair with the target site, which is having poly-T, and then from then on transposition occurs. Okay. So there are many examples. Until now, what we have seen are the strategies of different transposons. What the real examples are there will be little bit differences between them. I hope you remember that we have heard about TN5, TN10, TN7. They are different in the way the second or non-transfer strand is cleaved. You can look back in that. But I want you to understand that there are several examples. We will not discuss all of them. We will see a few, but I would encourage you to read at least this table so that you have good enough idea. But you are more than welcome to read the whole of this chapter because it will give you new ideas. Transposons are important genetic tools that are used in reprogramming, in stem cell biology, regenerative medicine, and many other recombinant DNA technology, and so on. For that purpose, it's a good idea to read this part of the chapter very well. So we'll talk about one of the examples, that is TN10 transposon. So it is about 9 kb, 9,000 base pairs. It is autonomous. Why are we calling it is autonomous? What do we need for one transposon to be autonomous? It should have inverted repeats, and it should have transposase G. Let's look at this. It has inverted repeats, and it has a transposase G. It moves from one place to another place in cut and paste by hairpin mechanism. I hope you remember, after the first strand is cut, it forms a hairpin like this, and then later it will open up again and then gets integrated into the target site. What is important here is the complexity that is involved is that it is a composite transposon. Composite means it's a mixture of transposons. Why are we calling that? If you can see, from here to here, it is one transposon. From here to here, it is another transposon, right? IS10 left, IS10 right. If you count from here to here, if you're just considering that this is one, sorry, this has one inverted repeat, and this is the other inverted repeat, then you have another transposon. Okay, so just look at IS10 left and IS10 right. They both are absolutely same, okay, sequence wise. The difference between IS10 left and right is that left has a defective transposase. What does that say? It is something like it is non-autonomous. If you just consider IS10 left, non-autonomous, whereas IS10 right has a functional transposase gene, so it is an autonomous transposon. You can also count IS10 right as the helper transposon, because the transposase that is produced from this gene can also be useful for the transposition of IS10 left. Okay, I hope you are following it well. So it is a composite transposon because it has three different transposons. So this is one. I'll just probably write the numbers. 1, this is 2, and if there is a transposase that is produced and it has, say, 2 subunits, one of it is binding to this inverted repeat and the other one is binding to the extreme right inverted repeat, then this whole thing can move from one location to a different location, and that's why you call it as it is a composite transposon. The importance or biological significance of it is that it also has tetracycline resistance genes. So these are some genes. Say, for example, if we have a bacterial infection, and to kill that infection, we give antibiotics. And those antibiotics, something like tetracycline, we will learn about antibiotics when we read the lesson on translation. It is an inhibitor of translation. So if we are giving tetracycline can go here and kill the bacterium. But over the time, what happens is there will be some mutants. Somehow they might have gained a gene, and that gene will produce, say, for example, this is a tetracycline gene. It produces an enzyme. And if tetracycline enters in, the enzyme will destroy it. And now the bacterium is resistant to tetracycline. Okay. So when you will also learn, when you're trying to think about different problems, scientific problems that we have, real life problems. One of it is the generation of antimicrobial resistance. You will find that majority of the bacteria are gaining resistance very fast. That means all the antibiotics that we have, tetracycline, chloramphenicol, many other antibiotics, they become useless to us anymore because the bacterium is now resistant to that particular antibiotic. That means you have to generate so many new antibiotics. Only then you will be able to treat the bacterial infections. So that's not the only point what I'm trying to describe here. Let's take a bacterium. Okay, it has its own genome, and assume that it has the transposon TN10. I'll just write TN10. And think of this bacterium also has a plasmid. Okay, and transposon can move from anywhere from one place of a DNA to another place of a DNA or to a different DNA. If the genome and the plasmid are within the same cell, TN10 transposon can move on to this onto the plasmid. Think of it that way. Say, for example, usually you will have about 10 copies of a plasmid, or sometimes even more, sometimes less. It depends on the plasmid. And say, for example, this plasmid died. What would happen? It will release all the plasmids that it has. Okay? And these plasmids can be taken up by another bacterium. Plasmid will enter in. And the plasmid has, sorry, the new bacterium host has its own genome. And the TN transposon can then move from the plasmid to the genome. That means in this way, why people have actually studied a lot about TN10 transposon is because it is associated with tetracycline resistance genes, right? Because the ability to move has made transposons a threat to us in one sense. But yeah, this is just one example of what the direct implications of transposon on our daily lives. And we have transposons in our own body too, in our own genomes as well. Just give a second. Still loading. Okay, it's going to crash. I will restart that again. So the other one is there are several ways of how they regulate their genome, their copy numbers within the cell. I'm not going to describe about it, but you should know one thing, that the number of transposons. Why should there be a copy number control is also something you need to discuss. I'm trying to put the genome. This line represents, or say this represents the genome of bacteria, and there will be different types of genes, right? Some genes are essential, some genes are not essential. So here is the genome. Well, I think I want to take a little bit of time and explain you a concept. I hope I will get to understand. I will then understand. This is a story, and you're supposed to participate and try to answer. In World War II, there was a fight. I don't know if I discussed this with you or not, but anyway, I will do it, redo that. So there were in World War II, there were fighter jets, and they're expensive instruments, and they can be shot down by enemy flights, right? And so one of the admirals thought to do something about it, to make a scientific study and see what we can do to protect our airplanes, because the pilot is lost and also the plane is lost, which is expensive, but they're also crucial for the fight. So there was a place, and these flights would go to the location, drop the bombs, and then they have to return to the place. Say about 100 of these flights went ahead, and about only 50 returned. So we are losing about 50% of the flights. So they wanted to protect the flight, but then you cannot put all the iron sheeting all over the body, because then it is too heavy. If it is too heavy, it cannot fly or it cannot be in the air for too long. It will consume too much power. So we want to armor the aeroplanes to reduce the loss, but we don't want to armor it all. Okay, so the several statisticians were and scientists were asked to do what, and then they analyzed. What they found was they found a lot of bullet hits on the wings and maybe fuselage, but not so many on the engines. Say, for example, assume that this is an engine. Assume that this is the. I'll just put it as here. Okay, we'll just put. Say this is the engine of it. There are a lot of bullet hits on the wings, but not so many on the engine. So they analyzed, and I think his name is Wald. He suggested. And now we have. Okay, I will ask this question. We have to armor the flight. Where should we arm? Where should we use the protective coverings on this aeroplane? You're supposed to answer. Try something, everybody. Yes? Jayashree?
Yes, sir.
Yeah, answer. Where do you think we should protect? The wings, fuselage, or engine, or tail? Yes? Which one should we answer? Which one should we cover? I want you to answer this. Okay. So typically, if we are thinking in what we see, we would be tempted to have a covering on these because that is where majority of the hits are, right? But that's not what the statisticians suggested. They suggested armor the engine. Why is that? All these 50 flights that returned had these bullet wounds, which would mean that even though they are hit on the wings, they were able to return to the base. You never find an airplane. You don't find an airplane that has serious damage to the engine because they never make it home. They die out. They sink in between. They crash. Okay, so this is something like evolutionary thinking about. So it will be half a thinking if we seem to just focus on, okay, we have so many of bullet wounds on the wings, so we'll just cover this up. That's not critical thinking enough. We have to think a little more beyond whatever exists is tolerated. If the holes are there, they are still able to they are tolerated. But if you don't find a hit on engines, if you don't find flights with hits on engines, that means if the engine is hit, they don't come back. Okay, so I hope I gave you something. You can go to YouTube and try to find out this story, or you'll have a better perception on that. So what I'm trying to say is the same thing here. If there is a transposon and here is a genome and you see a transposon here, okay, this transposon can move to any of the genes. Often you will find that the transposon is in the intergenic region and never in the never within the coding region of an essential gene. What would happen if it goes into an essential gene? Then this gene becomes mutated and it becomes non-functional, and then this whole organism would die. So in all the organisms that we see, majority of the cases, what we will see is the transposons are in non-essential regions. That means if it hits a essential region, it will die. The organism will die. The cell will die. It's the same thing as what I was just describing about the aeroplanes. So if a transposon is moving from one place to another place continuously, this one goes by cut and paste mechanism, right? TN10 goes by cut and paste mechanism. Say, for example, it moved here, no problem. It moved here, from there to here, no problem. If it moved from here to here, then the cell dies. Okay? So that is how you should think. If a cell is existing with certain mutations, that means those mutations are not good enough to kill the cell. So in genomes, you have some genes are essential and some genes are not essential. Well, you will get to that. You'll be able to understand, consider this as a kind of primer for you to think at the level of genome as a system. So their copy number is usually tightly controlled. When we describe about gene regulation, we will try to have a look at these. Right now, it's too early because translation, transcription, many things haven't been discussed yet. So there is also, if you had a genome and there is one transposon and it is moving by cut and paste mechanism, how many genomes will it, how many do you expect in that genome at any given time? You can only have one, because it is the same thing that is cutting itself, moving to a different place. From there, it is jumping to a different place and so on. But you would find that some transposons like TN10, you can find more than one copy. And how does that happen? Here is a nice way of seeing it. During replication, so this part of the genome is replicated, you have two transposons, right? Now what happens is, by cut and paste, it will move, the transposon will move from here to this location. You have a double strand break, and by homologous recombination, this will form here a holiday junction, and it is copied to this location as well. So now you have two different transposons. The other one here, it is going to get replicated and it will form two as well. So the genome, the actual genome had one copy. Because it is forming replication, it is going to generate two different DNA molecules, and each one will get one. But that's not what is happening. Each one are going to get two TN10 transposons, because if it is associated with replication, that is what it can increase in number. Yes. So there are lines, long interspersed nuclear elements and short interspersed nuclear elements. You will find we have read about it a little bit when we are reading about the genomic organization. And these are the ones that belong to the poly A retrotransposons. What do poly A retrotransposons have? 5' untranslated region, 3' untranslated region. You have two genes, ORF1 and ORF2. And we also have poly A tail. They are between 5 to 11 adenines. Okay, this is an example of poly A retrotransposon. We also described this thing about pseudogenes a little while ago. Sometimes, what are pseudogenes? Upon transcription, they get spliced, mRNA gets spliced to form a mature mRNA, which we will study in 2 lessons now. So just take it as an introduction itself, only as introduction. And we have learned about retrotransposons. That means they have this reverse transcriptase enzyme. If a mature mRNA, host, sorry, the host gene can be reverse transcribed by the reverse transcriptase to generate a DNA and then put it into the genome as if it was a transposon, right? And then that is how pseudogenes actually form. It is the activity of the reverse transcriptase enzyme of the transposons that carries out such an action and is responsible for the generation of pseudogenes. Okay, from here, we are going to talk about antibodies and the recombination that is involved. So if you don't understand, it is all right. Just try to know a few things. Antibodies have 4 chains, 2 light and 2 heavy. The ones that are shown in blue here are the heavy chains because they are larger in size. And then you have these that are light chains because they are smaller. And there are disulfide bonds like this. I hope you know what is a disulfide bonds. This is a quaternary structure of how an antibody is. And this is the region that binds to the antigen, right? You have 2 regions that bind to the antigen. And those are the variable regions, in the sense that if you take 10 different antibodies, all of them will have this region same. And that's why it is called as constant region. Whereas these 10 different antibodies will differ in this region. That is why it is called as variable region. Okay, constant region, variable region. So if you look at the light chain, you have some constant region and you have some variable region. And the tips of these are called the antigen binding sites, which are hypervariable sites. So there are B cells and T cells. B cells secrete the antibodies, and antigen is anything foreign to our body. If a protein or a molecule that is large in size enters into our body, that is called an antigen. Anything foreign to our body that can be recognized by immune system is antigen. And there are a large number of antibodies and so assume that this is a body, right? An individual. The whole universe is around, and you have billions and trillions of molecule types, and any of them may enter into the body. Okay, if they entered into the body and somehow this body has immune system to remove or destroy that foreign antibody. How many molecules we are talking about? Billions and billions of cells here. Molecules outside, external to the body. Any of them may enter into the body, and the immune system, in most cases, is able to thwart that attack. That means the antibody should be specific to that particular antigen. So if you had these many molecules, you should have had antibodies with so many types of antibodies that can recognize those molecules. That is where generation of diversity is very important. So you can have the same antibody, but with a different antigen binding site. And that's why we want many, many thousands of these kinds of antibodies. What is an epitope? Say, for example, there is a protein that entered into our body, and the protein has several structures like this. And antibody can only recognize one recognize a small part of a protein. It cannot recognize the whole protein. And if the antibody can recognize that small part like this, that part is called as the epitope. Epitope is the part of the antigen that is recognized by a particular antibody. Okay. I hope I gave you some introduction to about antibodies. We talk what you have to remember for understanding this particular part of recombination is that the antibody, the light chain. Okay, I will just put the light chain a little, only small one, and then let's see. I will put it this way, linear, and this light chain has two different parts. If you look at it, this part is the constant region, and this is the variable region. If you see, even there is a small junction between this variable and the constant region, which is called the joining region or J region. So it should have a variable region, it should have a joining region, and it should have a constant region. That means they should come from different parts of the genome. So how diversity is generated is the question. And there is a lot more biology than what I can discuss in this particular class with regard to how immune system works and so on. I hope you remember, you carry this information to your next course when you learn, where you learn about immunology, and that will be very useful to you. So in the germline, in every cells, we have several B regions. This is the B region, this is the J region, and this is C. We have several genes for V. We also have several genes for J. Sorry. And we have a few regions for C. The constant regions will be mu, delta, gamma, epsilon, alpha, and so on. And variable regions, you have so many of those. And in other ones, you also have D region, and in addition to VJ. So when the recombination occurs, we say in the first one you're seeing here, we'll say recombination happens between V regions and J regions, like in here. In some others, we will, in the heavy chains, you have B, D, and J. So that's why they put the D in the brackets like this, VDJ recombination. So what happens is, when the B cell is differentiating and it is matured, it will lose many of its V regions. And eventually it will have, say, for example, this is the transcribed. V region is recombined. V3 has recombined with J3, here like this. The region here is lost. Okay, let me try once again. So if you see, look carefully what is happening in here. Recombination, site-specific recombination has happened between V3 and J3. All this region between is lost. Okay, so you see this V3 continuous with J3, and that is transcribed. Then you have splicing. During splicing, you will have this C, the constant region, coming up, and now you have continuous open reading frame. Start codon will be here, stop codon will be here. And that, upon translation, will produce a light chain containing V3, J3, and a constant region. Okay? So what we are interested, right, in the next chapters on transcription and splicing, we will learn these two steps. How does transcription occur and how splicing occurs? In that, we will learn about alternative splicing and many other stuff. For this particular lesson, we are we are focused on recombination, and recombination occurs here during differentiation and maturation of B cells. Well, this is the heavy chain and so many other things. I don't want you to, I don't need you to mug up all these things, but I hope in the next course, when you're reading about immunology, you will be able to relate the recombination process with, with the generation of diversity in antibodies. So how recombination happens is there are, so I hope you remember, again, try to focus on this. This is for large heavy chain, VDJ. If it is small one, you will have V and J. There are certain sequences, something called here represented by blue triangle, which is having which is called as recombinational signal sequences. Don't confuse it with recombinase recognition site we learned in the earlier part of this chapter. These are RSS, recombinational signal sequences. So each of them has its own structure. And that structure is, on one side, it has a 7-mer, there is a 12 base pair spacer, and on the other side, you have a 9-mer. 9-mer means 9 bases, 7-mer means 7 bases. And look at the 5' side of J chain. You're finding, you will see that it has a 7-mer, 23 base pair spacer, and a 9-mer. So the recombination always occurs between 12 base pair and 13 base pair spacers. It cannot occur between two 12 base pair spacers or 12 or 23 base pair spacers. It can only occur between a 12 base pair spacer and a 23 base pair spacer. I hope when you look into the next slide, you will understand that. We will just go through the mechanism first, and then we will try to have a look at that. Okay, it's not here. But remember this, that the recombination can only occur from this. I will try to use this slide. So the green one is a 23 base pair spacer, and you have a 12 base pair here, and VDA has 12 base pair, and J has a Can you hear me? Sorry, lost the connection.
Yes, sir.
Okay, thank you. So I was trying to describe, I said, this is one of the rules that you should remember. Base pairing recombination always occurs between 12 base pairs and 23 base pair spacers. This V here has a 12 base pair, or this is 23. The blue one is 12, and here is 12, and here is 23. So if recombination has to occur, and they have to be inverted repeats. This is another rule. These are the two important rules that they have. One is recombination can happen only between 12 base pair and 23 base pair spacers, and they should be inverted repeats. So the question is, can recombination occur between this and this? Yes or no? Yes, it can occur because it is two different. One is 23 and the other one is 12, and they are inverted repeats, so this is a go. Let's see about this with this one. One is 23 and the other one is 12. They are fine with this rule, but they are not inverted repeats. They both are pointing in the same direction. So this is not satisfied. So recombination does not occur there. Let's see if recombination can occur between this and this. They both are 23 base pairs, so they cannot occur. Let's try with this one. I'll try to use a different color. Let's try this 12 base pair with this one. They are inverted repeats, but they both are 12 base pairs, so that will not occur. This with this one, yes, they cannot. They are two different ones, but they are same. They are not inverted repeats, so that does not occur either. Okay, I hope you understood that. I will try to retain what is possible and erase the others so that we can continue with the other ones. So this is not possible. We said these three are also not possible, and we also said this is not possible. I've retained what was possible here. And now let's see. So this only has one possibility to base pair recombine with this 23. Let's see this one. This one cannot base pair with this. Sorry, this cannot recombine with the 23 base pair one because it's in the same orientation. This can recombine with this one because one is 12 and the other one is 23, and they are inverted, so it can happen. Imagine what would happen then. You would always get only V, D, J in the order. If this is the setup, you would never get something like VJ. Why is that? Because of the arrangement of the recombinational signal sequences. Okay? So it is an interesting and important area to understand about immunology, antibodies, and everything has a molecular basis. If you know a phenomenon and you don't know the molecular basis or the genetic basis of it, then we didn't understand it properly. So I will just stop with this. There are two proteins called as RAG1 and RAG2, which are the ones that make the connections, recombination between this and this recombination signal sequences. So RAG1 and RAG2, and also there is another HMG protein. They act something like transposases, something like they are not transposases, and they make a cut, and then they make the second cut. I hope you have identified this way in transposon non-transfer strand cleavage. In some, the transposon forms the hairpin like this, and the host DNA is like this. In other cases, the host DNA forms the hairpin, and the transposon is like this. I hope you remember that. Third category, when we were talking about the three different ways of how this non-transfer strand is cleaved. So this one follows this hairpin formation of the donor site, and then they will eventually, this part will be discarded. It forms into a circle like this, and it will be discarded. Whereas the recombination between these two will occur, and now you have a continuous VJ. This is for light chain, so we are not talking about D region. We are only talking about VJ, right? And then further recombination can occur because you also have to recombine with the constant region. Okay, this concludes this lesson. Try to on transposition or site-specific recombination and transposition. Please try to read it. It's an important lesson for your thinking about genes and genomes, how genetic information can be modified. Okay? We'll meet in the next class. We will start on transcription. Okay? Take care and have fun. Bye bye.
Thank
Thank you, sir.
See you. Bye.
thank you, sir.
See you. Bye.